The procedure visualizes the hyperproduction of CAG type 4 secretion system pili, which occurs under conditions of iron limitation. First, culture H. pylori under conditions of iron limitation. The second step is to co-culture H. pylori with gastric epithelial cells. Next, process the co-culture samples for scanning electron microscopy. Ultimately, high-resolution scanning electron microscopy is used to show increased CAG type 4 secretion system pili production under conditions of iron limitation.
The technique in this manuscript is important to H. pylori-related diseases such as gastric cancer because the CAG type 4 secretion system is associated with increased risk of negative disease outcomes. Thus, a better understanding of the regulation of this important bacterial surface organelle could reveal novel chemotherapeutic targets.
And the advantage of high-resolution scanning electron microscopy versus existing methods such as fluorescent microscopy is that individual type 4 secretion pili can be visualized.
Individuals new to this method of electron microscopy have problems with the procedure, specifically the wet chemistry, as it requires careful timing and a delicate hand.
On blood agar plates, grow H. pylori PMSS1 bacteria and its isogenic CAG-E mutant, which lacks a functioning type 4 secretion system. Inoculate cultures in modified Brucella broth, supplemented with cholesterol, to grow overnight.